Hi team, in this video I'm going to be talking about topic 3.1, which is called genes and chromosomes. This is probably going to be quite a long video. I'm going to try and go through the whole topic in one video, so you'll need to pause regularly to make notes. There are various opportunities to pause and do mini activities throughout the video, but at any point, if you want to pause, then you can feel free to do that and make decisions about what kinds of notes you want to make. So first of all, this is what we're going to cover. We're going to look at the terms gene and allele. So we're going to look at what they mean separately, and then we're also going to look at how they relate to each other and how they're different from each other. And then we're going to look at how alleles are formed and how they're different. A lot of this knowledge you might still have from GCSE. If you remember it, that's great, and you might be able to watch this video at a faster speed if you're finding that you keep up. If you're finding that this is hard, don't worry. I'm going to come right in from the ground up. So you don't need to worry if you can't remember. It just might take you a little bit longer. Once we've had a look at those understandings, we're going to apply them to some new contexts. So we're going to look at the exact cause of sickle cell anemia and what its consequences are. You already have compared the number of genes in humans with other species, but what we're now going to do is use a database to compare the same gene in two different species, just to see how similar and how different they are. So let's begin, and first of all, let's talk about what a gene is. So you should have already written down 3 bullet points of what you think a gene is, and we have studied DNA and nucleic acids quite a lot fairly recently, so hopefully you've got some ideas of what a gene is. A gene, kind of the idea of a gene, has 3 main parts to it. So first of all, a gene is a heritable factor. Heritable means that it can be inherited from one generation to the next. So in humans, you get it from your parents. Genes can also be passed horizontally in bacteria, but basically it's something that can be passed from one organism to another. It consists of a length of DNA, and we've looked in a lot of detail about the structure of DNA, so you know what I mean when I say it's a length of DNA. A lot of people say, how long a bit of DNA? And the answer to that is, how long is a piece of string? A gene influences a characteristic, and it does that by encoding one protein. And we've looked at the processes of transcription and translation, so you know how a gene encodes a protein, and the length of DNA is just however many DNA bases are needed to encode all the amino acids for that protein. So some genes are a couple of thousand base pairs long, some genes are a couple of million base pairs long. It depends on the protein that they're encoding. So different genes have different lengths, and different chromosomes have different numbers of genes on them, but we'll come to that in due course. So first of all, you might have seen this slide before. It might be a little bit familiar from GCSE. If you feel confident in skipping, then feel free to. And if you are not confident in skipping, then please watch it. All right, so all humans have the same genes, but why are we different? Let's take an example. I've got blue eyes. We've all got genes for eye colour. We've all got genes that encode the protein that makes a pigment that makes your eyes a certain colour. So why do we all have different coloured eyes? Why do we all have different coloured skin, different coloured hair? Why are we all different heights? And so on. And the answer is yes, we've all got the same genes. We've all got the same number of genes. We all make a very similar number of proteins, but we might have different versions of those genes. So you might already know that the original humans, the very first humans, were thought to have brown eyes, and that's because they had a gene that encoded a protein that gave them brown eyes. But since then, humans have evolved different colours of eyes. So you could get brown eyes, hazel, you know what eye colours are, I'm not going to tell you. Each of those different versions of a gene is called an allele, and that's A-L-L-E-L-E-S. An allele is a version of a gene that has a slightly different base sequence. Okay, so people who have blue eyes still have an eye colour gene, it's just that their gene has a slightly different sequence of bases on it. Similarly, the first humans were thought to have very dark skin. They had a lot of melanin pigment in their skin, which protected from the sun, because the first humans lived in Pangaea, which was an equatorial area. I'm sure you'll learn more about that in other subjects. But again, over time, changes in DNA sequences occurred that caused new and different colours, shades, or depth of pigment in people's skin. And the cause of that is by mutation. So a mutation is a change in the base sequence of a gene. It can happen because of radiation, for example, from the sun. It can happen because of certain chemicals, particularly we're told about the ones in cigarettes. And also you can inherit a different pattern of DNA from your parents, which we already know about. So every allele is produced by a small change in a DNA sequence. And the consequence of that is that a different protein is made, which has a different shape or a different function. So just to check that you were listening, just to check that you were paying attention there, here is a quick question, and I just want you to write down the answer to this question. What's the difference between the alleles of a gene? So just take a note whether you think the answer is A, B, C, or D. And at the end of this video, I'm going to ask you to send me a message with all of your answers so I can check how well you followed the video. Okay, so if you want to pause now and have a bit of a think about that question, otherwise we will carry on moving on. So now we've looked at what a gene is and we've looked at what an allele is, and we know where alleles come from and how they're different. So now it's time to look at an application, a specific example of genes and alleles, and comparing the different versions of genes in different species. So you might recognize this. It's a picture of a red blood cell. The fancy term for the word red blood cell is erythrocyte, which is spelled E-R-Y, and now I can't remember the rest, T-H-R-O-C-Y-T-E. The suffix cyte means cell, so erythrocyte is red blood cell. This red blood cell, you can see, is a disc shape with a flat part in the middle. That shape is known as biconcave. And as you know from previous lessons and from your previous biological learning, it contains a protein called hemoglobin, and hemoglobin picks up oxygen and carries it around your body, delivering it to tissues. So hemoglobin is a protein. In fact, it's 4 proteins, and those 4 proteins are therefore encoded by genes. And funnily enough, there can be a mutation in genes. This image shows a sickle red blood cell, which is clearly the wrong shape. So what's happened here is there's been a mutation which has created a new allele of the hemoglobin gene. That hemoglobin gene has therefore transcribed and translated and produced the wrong sequence of amino acids. We're going to look in more detail about the specifics of that in a moment. But the thing that I want you to notice is that obviously this red blood cell is completely the wrong shape. For that reason, it makes it very hard for it to pick up oxygen at all. Also, because it's kind of spiky, it tends to get clumped together, and it tends to form blood clots quite often. So people who have sickle cell anemia often suffer from blood clots more frequently. It's also a lot less flexible than the normal red blood cells around it. So that means it finds it harder to get through the capillaries. So people who have sickle cell anemia find it hard to get oxygen to their tissues, and they also often suffer from blood clots. Now, I'm about to show you the mRNA sequence for normal hemoglobin and the mRNA sequence for sickle hemoglobin. I'm not going to show you the full sequence. I will show you that later. We're just going to look at the first 12 codons on the mRNA. So here are the first 12 codons of the normal mRNA. And here are the first 12 codons of the sickle mRNA. So you can just pause the video here and have a look and see if you can spot the difference. So you should be able to see here that this sequence reads GAG and this sequence reads GUG. So this is the 1, 2, 3, 4, 5, 6, 7th sequence. It's the 7th codon on the messenger RNA, and it's been swapped from GAG to GUG. So that means that this DNA sequence, the normal DNA sequence, is CTC, and the normal DNA sequence of the 7th triplet on DNA has swapped to this would have been CTC. Okay, so because the 7th triplet of bases on the DNA changed, that meant that the 7th triplet on the messenger RNA has changed, which means that the 7th anticodon of the tRNA will change. And so this starts to have a knock-on effect. Now you can see here, if you had GAG as your mRNA codon, so GAG, this would have encoded the amino acid GLU, that's glutamic acid. Because of the mutation that changed the DNA triplet, that then was transcribed to the wrong mRNA anticodon. I hate when that happens. The wrong mRNA codon, and that codon was switched to GUG, so GUG, and that encodes the amino acid valine. So it's only that one change, that substitution from a T to an A on the DNA, which changed an A to a U on the messenger RNA, that one substitution of one base on the 7th codon has encoded a different amino acid sequence. And just that difference is the difference between a healthy red blood cell and a sickle red blood cell. Now, just to really show you the magnitude of this, I'm going to show you the entire coding sequence. So out of all those bases that encode hemoglobin, just this one base is different. And as a result of that one base being different, the entire red blood cell doesn't function properly. So just to check that you've been listening again, what I'm going to ask you to do is to just try and figure out what is missing from each of these sentences. So pause the video here and have a think about what words go in each of these sentences. If you paused the video there and you're feeling completely stuck, so you've carried on hoping that I'm going to give you the answers, then what I will do is give you some help in terms of the missing words. Now, I haven't included the HL missing word, I've only included the SL missing word. So maybe you can figure out what goes where. Now, just keep a record of the words that you've put in what order, and keep that with the letter that you gave for the last question, because you're going to send this to me when you get to the end. Okay, so we've looked at the terms gene and allele, and you know how alleles are formed and how they're different from each other, hopefully. You've looked at sickle cell anemia, and we have compared the number of genes in humans with other species. So now we're going to do one final thing, which is to use a database. Now, I've said this to students in the past, and I'm going to carry on saying it to students now. I don't really know how the exam board would expect you to use this. I don't really know how they would ask you a question that required you to use a database, since you take the exam on paper, but I'm going to show you how to do it anyway, and then whatever they come up with, you'll have already experienced it. So what you're about to do is you're going to go to a website, and you're going to look at a gene called cytochrome C. I've picked cytochrome C for a reason. It's because it codes for an enzyme, and the enzyme is used in respiration. And as you know already, respiration is carried out by every living organism. So that means that this enzyme is also shared, or a version of this enzyme is shared by all organisms. Because it's so common, it's been sequenced in a lot of different species. So we have the sequence and can compare its length and position and the actual sequence of bases. And because it varies quite a lot from one species to another, we can actually use it to group species. It's been used for classification purposes. So organisms that have very similar cytochrome C sequences are closely related to each other, and organisms or species that have very different cytochrome C sequences are distantly related to each other. And also within a species, if there's like a mutation of the cytochrome C gene, because it's for respiration, it can be quite debilitating. It has a very negative effect, which means that generally all the members of a species have the same sequence. There's only one allele that survives, essentially. So what that means is it's identical within the species, and then the differences between species can be used to work out how closely they are related. So if you're feeling confident, you could ditch the video here and you can go and have a try of doing this by yourself. But I'm going to show you how to find out about a gene using the NCBI website. NCBI stands for the National Centre for Biotechnological Information. You don't have to remember that. I'm just telling you what it is. It's a really reputable, it's based in the US, but it's used internationally. It's contributed to internationally. Anytime that any information is published about biotechnology, anytime that a new gene is sequenced, blah, 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 loads and loads of things, it goes on the NCBI website. And it's accessible, public access. So it's one of these brilliant science things where scientists come together from all over the world and work together to find new information, which is then publicly available, shared with everyone so that everyone can learn and have knowledge. That's really, really good. Anyway, I'll shut up now. I'm going to go to this website. Okay, and in the top, it says gene here. You can find out all kinds of stuff. But it could get a bit scary. So let's stick with gene, and you're going to type in the search box, cytochrome C. Oh, I spelt it wrong, so this is a bad start. Okay, now you'll get a list of all loads and loads and loads and loads of cytochrome C genes. The ones that you're going to look at, you're actually going to look at the somatic Homo sapien one. So I'm not going to show you that because I'll give it away. And then you're going to look at the house mouse, Mus musculus. So I'm not going to show you that, I'll give it away. I'm going to show you the Norway rat. So I'm just going to go through the process of finding the information with the Norway rat. And don't worry about remembering these steps because I've written the steps on the doc. So first of all, you click on this link on the left-hand side, which is not super obvious. And then this will open up all of the information about this particular gene. The first thing you can see, if you look in genomic content, you can see its location. So the location in the Norway rat of this gene is 4q24. Now you're going to do more about what this means next lesson, so try not to worry too much. But 4 means chromosome 4, Q means the long arm of the chromosome, and 24 means gene number 24 on that arm of that chromosome. So you can find out its location. That's quite easy. And then if you go down to genomic regions, transcripts and products, which is utterly terrifying, don't worry about that. Go to this link on the right-hand side that says GenBank. And this has all the information just about this one gene. It's super cool. So here you can see that this gene is 2,101 base pairs long in the Norway rat. Might be different in the species that you look at. And then when you go down here, and I don't expect you to count them all or read them all, but this is the entire sequence. So this has been sequenced using dideoxyribonucleic acid, to those of you who've studied it. And you can see here, like this is the codon or the base number. So it goes all the way down here to 2,101. So you can have a look at that. Now, I'm not going to ask you to compare the sequences. I just want you to look at them and appreciate the magnitude and the amount of work that has gone into finding out this information. And you can compare length and you can compare locus or position between Homo sapiens and Mus musculus. And that, once you've done that, is us done for the day.